Good morning everyone. So today we will be talking about self-assembly and its application for tissue engineering scaffolds. So before we start with what self-assembly is, in this overall lecture we will be looking about what self-assembly is, what are the interactions that facilitate this self-assembly process, and how do we use them for our tissue engineering applications. So I will be giving a definition of two things today, what self-assembly is and what self-organization is. So self-assembly is the spontaneous associations of molecules under equilibrium condition into stable, structurally well-defined aggregates by non-covalent interactions. So just as a question, so everyone knows what a covalent interactions is and what a non-covalent interaction is. So today in self-assembly we will be talking only about non-covalent interactions, but why there is a definition of what self-organization comes across is, these two terms are interchangeably used in the biological process. So people come across both self-assembly and self-organization, but there is a very narrow difference between what self-assembly is and what self-organization is. So when we look at the definition of what self-organization is, it is a process in which order is attained from a disordered state, similar to what self-assembly does, but here an external energy is input into the state to maintain the equilibrium. But whereas in self-assembly there is no energy that is given to the system, it is a spontaneous process. So self-organization comes up into other macromolecular systems, where today we will be dealing only with self-assembly. So self-assembly comes into a field of supramolecular chemistry. So I am not going to bore you with more of chemistry terms today, just a little bit of how chemistry and biology are interrelated, we will be looking into it. So supramolecular chemistry is defined as chemistry beyond the molecule. So how do we say that molecules come up? So we have an atom, and then these atoms are covalently bounded to form a molecule. Say you have your C double bond O and the NH molecule forming an NH2. So this is forming of a one molecule. So this is what the normal chemistry involves. But what goes into beyond the molecules? When these molecules come together, what happens? What is the aggregates that are being formed? When you look at the diagram there, you have a molecule that is a pictorial representation. So this we say the initial supramolecular chemistry was termed up with host-guest interactions. You take one molecule to be the host and another molecule to be the guest, and then when they all combine together, there is an interaction that is happening in between, and then these form the aggregates, which we called as a supramolecular aggregate, or a supramolecular chemistry is the term overall. So unlike the covalent bonds that are being formed within the atoms, here there are only non-covalent interactions that come into play. Brief history of how this came across. So we all know how this LB film is, your monolayer adsorption film that we have all studied. So this is how an amphiphile or an oil displaces into the water. So this was a monolayer assembly initially, that was in 1774 by Benjamin Franklin. And then there was another breakthrough in 1891, which is the cyclodextrin molecules. So the image that you see there, the 1891 image, it was found by Villiers and Hebd. So these cyclodextrins are the most conventionally used systems for host-guest chemistries. So what is a cyclin or what is a cyclodextrin is, the common polymer that we see, the glucose polymer, is all linear in nature, but this was the first cyclic polymer that was found. So these cyclins were of three different classes, the alpha cyclodextrin, the beta and the gamma, and this depends upon the different sugar units. The number of sugar units is the one that is varying. And then there was another thing that they found was the substrate enzyme specificity that we all see in the biological system. So we have an enzyme, we have a substrate, there is a very high complementary binding that happens between an enzyme and a substrate, because of which they bind and then the product is formed. So this is one level of an host-guest system that happens, which gives you a product formation within an enzyme and a substrate. And then we have the Watson and the Crick model, that is the DNA, which is the basic model of our human body. So what runs across is the double helix model. So you have two different parallel chains running across, and then you have the hydrogen bonding within them. So this hydrogen bonding is a non-covalent interaction that keeps this DNA molecule intact. And so these were the three people who were given a Nobel Prize for their supramolecular chemistry. So these three molecules, the crown ethers, the sperins and the cryptins, are all cage-like systems that was all formed, and these are organic molecules. This was one of the first ligand metal process or ligand and host gas systems that was initially synthesized, and this is when the field of supramolecular chemistry came into existence and people started noticing what supramolecular chemistry is or how we can utilize them for various other applications. So, before we deal of how we are going to take these self-assembly or what supramolecular chemistry into tissue engineering, we need to know what are the interactions that come into play and how they are different from the covalent interactions. So, the 5 major interactions that we will be talking about are the electrostatic interaction, the hydrogen bonding, the pi-pi interaction, van der Waals interaction and the hydrophobic effects. So, the first one will be electrostatic interactions. So, so all these interactions are not high energy involved unlike your covalent bonds. So, the covalent bonds will have around 500 kilojoules per mole of energy, and to break a covalent bond you need so much of energy that is also involved. But unlike interactions, electrostatic interactions or non-covalent interactions, we do not term them to be bonds, we always say them to be interactions. There is very less of energy involved in all these chemical processes and they are mostly reversible in nature. Just with a stimuli or any responsiveness, they are reversible, bringing them back to their original form. So, the electrostatic interactions is the ion-ion interactions. So, we know what an ion is, a cation and an anion with a positive and a negative charge. So, when there is an interaction between two differently charged molecules, we call them to be as an ion-ion interaction or an ionic interaction. And then what is a dipole ion interaction? So, what do we say as a dipole molecule? You have a polar molecule, and in chemistry we must have all learned that electrons move across and then there is a charge separated species that come across. So, what happens is there is a partial positive charge and a partial negative charge within the molecule that happens. So, when there is a partial positive and a partial negative charge, these ions are immobilized in different regions, and when this combine or interact with an neighboring ion or a dipole, then there is a difference in the interaction. So, we have a dipole ion interaction or a dipole-dipole interaction depending upon where the dipole interacts. And so, the major thing that we will be looking about is hydrogen bonding. So, this is one of the most important driving force that takes this interactions or the non-covalent supramolecular self-assembly taking forward. Because for tissue engineering applications we use very important biological molecules which all have an amide backbone or an NH group which is involved in that of the hydrogen bonding. So, let us first understand how an hydrogen bonding works and how you identify where the bonding happens. So, this results from the attractive force between an hydrogen atom covalently bonded to a very electronegative atom such as nitrogen, oxygen or fluorine with another very electronegative atom. So, let us take the conventional unit system that we take for defining an hydrogen atom, the water molecule. So, in the slides you see two water molecules, one to your left and the other to your right. So, there is a partial bond that is being formed in dotted lines between an O and an H. So, there is the oxygen which has a lone pair of electrons. So, that is the highly electronegative atom, and then you have an hydrogen which has a partial positive charge. So, any hydrogen bonding that you take, you need to identify what is an hydrogen bond donor and what is an hydrogen bond acceptor. So, the hydrogen bond donors are always the hydrogen which is linked to the electronegative atom. So, in this case the right hand hydrogen which is linked is your hydrogen bond donor, whereas the most electronegative atom is your hydrogen bond acceptor. So, any hydrogen bonding that you come across, try to identify where your donor and your acceptor systems are to know whether these molecules can undergo an hydrogen bonding, because any hydrogen cannot have an hydrogen bonding. Only if it is linked to an electronegative atom, it can facilitate an hydrogen bonding. A CH bond will not give you an hydrogen bonding, only an OH bond can give you an hydrogen bonding. And then we have the pi-pi interactions. So, these pi-pi interactions are within the pi molecules or the aromatic molecules that we always say. So, the pi electrons are always delocalized within your electron cloud in the aromatic systems. So, they are not stable, they are more in the electron cloud. So, when these molecules come into contact with close each other, they form a stacking kind of a nature. So, the pi-pi interaction is most commonly found in only aromatic systems such as your benzene units and then your phenylalanine, all your aromatic compounds. And this can have a difference in the arrangement where you have a face to face pi-pi stacking, or then the T-shaped packing is a face and an edge packing, and then you have a parallel displacement packing of the pi-pi nature. And then we have the van der Waals interactions, the weakest interactions among all of them that is claimed to be the van der Waals interaction. This is also called as the London dispersion forces, where people debate on whether both are similar or they have a similar property. So, van der Waals interactions, like how we saw for a dipole or an ion dipole, are not between polar molecules, are between non-polar molecules. So, we know that when there is a polar molecule, then there is a charge separated species that happens. But even in non-polar molecules and even at inert molecules such as helium might have these temporary dipoles or oscillating dipoles. So, when they are put in a medium, due to some probabilities, there might be an electron that is moving and you will have a temporary dipole. So, these temporary dipoles are always oscillating. So, you have a very weak partial positive charge and a partial negative charge. So, what happens is at that moment when there are similar molecules coming across to each other, so these partial positive charge. So, this is how the blue molecule says. So, you have a very negligible partial positive and a partial negative charge in the non-polar molecule, and when they all come across, they start oscillating in a similar tune or a packing pattern, and then they form a self-assembled array. So, this is how a van der Waals interactions takes place. But when you orderize or categorize all the interactions, the van der Waals interactions are the least among all the non-covalent interactions. And then we have the hydrophobic interactions, which is found in all your protein systems, and then the hydrophobic cavity, how the protein starts folding. So, these interactions are mainly because when the non-polar molecules, when put inside water or an aqueous medium, how they react. Okay. So, what happens when you are put into a very unfavorable condition? You start to become very closed, you do not open up. So, this is what happens even when you put an hydrophobic molecule which does not like water inside the water system. So, what happens is they become very close, trying to minimize their energy, and this is what, so that is what the water surrounding the molecules form a cage around the hydrophobic molecules. So, because there is a change in all this, there is a rise in the entropy in the surrounding system, and the hydrophobic properties are being inside the interior buried deep. So, these interactions are very majorly found in your protein folding processes or your micelles and bilayer assembly, where they all interact with your water system. So, we have seen all the different interactions, that non-covalent interactions that take up all this. So, let us see how they drive our life. So, there are three important systems within the biological system that is well characterized and studied for all these non-covalent interactions. So, they are the proteins, the DNA and the lipid structures. So, the first one will be often hydrogen bonding in DNA. So, when you look at the DNA structure, you all know there is a DNA double stranded helix, as I have already told before. You know there is a phosphate group or a sugar and a nucleobase. So, the major thing that is holding the DNA double helix is the hydrogen bonding between the two nucleobases. So, we have an adenine and a thymine, guanine and a cytosine, and always the bond is between A and T, G and C. So, anyone knows why it is always between A and T and G and C? Why not is the other way?
Because the interactions won't be stable as a way.
Okay, what interactions? Okay, let us take an example. So, this I am giving you as your molecule. So, the common thing is adenine and thymine, and then guanine and cytosine. If you are going to link the other way, say you will have adenine and cytosine, guanine and thymine, how many hydrogen bonds each one will have? 2 for what?
Both.
Does everyone agree with this answer?
Where hydrogen bonds, it might not be planar.
But the hydrogen bonding is more of a distance related phenomena.
Yes.
So, when we look at adenine and cytosine, say for example, look at your structures. You have one donor and one acceptor system, and then whereas you have a cytosine, you have two donors, one acceptor, but they are all misplaced. So, when you have an adenine and cytosine, it will have only one hydrogen bond, and thymine and guanine, it will have two. So this is the reason there is always have to be complementary, that A and T will bind, G and C will always have, and that is the reason how hydrogen bonding plays a major role in keeping this double helix strand in place. So when we design molecules also, there can either be an intramolecular or an intermolecular hydrogen bonding. Within the molecules also there can be an hydrogen bonding, but for all these non-covalent interactions, the distance between the molecules plays a driving role. So there is a very important distance, because it can either have a repulsion or an attraction. So the distance between the molecules will help in facilitating the self-assembly process. And then we come across the protein folding. So here in protein folding, it is mostly of the intramolecular self-assembly system. So the first structure or the primary sequence of your proteins will be amino acids. These are all covalently bonded. So you have the amino bonds and then forming your long peptide sequence. And what happens when you go for your secondary structures? The secondary structures, the major role playing is the hydrogen bonding. So this forms your alpha helix and then your beta sheets. So after this alpha helix and the beta sheet, they form with the hydrogen bonding. Then comes your tertiary structure into play. So the tertiary structure of the protein is more about the protein folding or the intramolecular hydrogen bonding that comes into play. So what happens is, when you put them into their aqueous environment, the hydrophobic interiors go inside, so that minimizing the exposure of the hydrophobic cavities to the protein exterior. So outside will have your lysine residues, your cationic or your anionic residues. So this is how your protein folds, and there comes the hydrophobic interactions into play. And quaternary structures are all intermolecular hydrogen bonding, where there are two polypeptides that come into interaction, where all proteins do not have a quaternary structure. And so the host guest chemistry is one of the important mechanisms. So based on this enzyme catalysis reactions only, we have formed these cyclodextrins, pilarines, all these host guest chemistry molecules. So there is a very high specificity when you come for an enzyme substrate specific molecule, and then this also plays a role in your antigen antibody binding, where they are very highly complementary in each other to them. And then the binding substrate has very specific pockets or sites, for which you have to design molecules to go and link to them for having an effect. And then comes the lipid bilayer. So this is how your monolayer assembly starts. So we have an hydrophilic head and hydrophobic tail, and these molecules, when put in water, they start to self-assemble. So either they form a micelle or they form a bilayer. So the bilayer is when two hydrophobic tails come together and then they start packing, forming a bilayer, and then they might also have a vesicle kind of a thing forming with that of the bilayer. So similarly, our eukaryotic cell membrane is highly composed of these phospholipids, and in comparison with that of the phospholipids, there is also a very large probability of having the cholesterol in the phospholipids. So the cholesterol also has a similar structure and shape, and thereby this is how we have this inflow of small molecules and then the hydrophobic interactions with other molecules coming into play, because these cholesterols are within the phospholipid membrane managing and maintaining all the inflow of small molecules and other molecules. And this is also one of the reason why you say that hydrophobic molecules bind to your cell membrane, small molecules go through smaller diffusion process. All this is all governed by how the molecules are arranged and what is the bonding that they all go in. And so after all these interactions and how they are in our day-to-day activities, so the major role of how a self-assembly is in comparison with that of a non-covalent interaction is that it's this stimuli sensitive property. So to forming a covalent bond, there is a lot of energy involved, and to break them also that is there. But unlike in a non-covalent interaction, there is a very little stimuli that is needed, either temperature, heat or any pH sensitivity, to have an alteration in that of the assembly that we have been formed. So this is how the biological process work, and this is what we are trying to mimic in our tissue engineering. And then we have a tailorable biological properties and the nanostructures that we can form. So all these self-assemble or self-aggregate to form stable structures, and these structures are mostly in the nano regime, and they also have, even if they go apart the nano regime, they have a nanostructure. Say if you have a fiber, they have a width in the nanometer regime. So these can be used and tailored for several different applications based on how we want them to be used. And so then we will be talking about the self-assembled systems for tissue engineering applications, the peptide self-assembled system, the host guest chemistry molecules that are being used. I am just giving you some of the examples. There are actually a lot of literature reports which currently discusses about all these systems, and then the pi conjugated systems and the hydrogen bonded systems. So, the first one we will talk about an aromatic pi conjugated system. So, the figure here, so this paper is from a ChemCom paper, and you have an phenylalanine molecule. So, it is a diphenylalanine molecule. So, diphenylalanine or F-MOC conjugated phenylalanine are known to form hydrogels. So, these are small molecules. When put in water, they start self-assembling, they form an hydrogel. So, how do you visualize a gel is by an inverted method. So, you have a solution, and when you know the solution does not flow, you term them to be formed as a gel. So, these alanines are known to form gel, and what in this study they have done is they have conjugated an indole moiety. So, the left one is an indole moiety to that of a diphenylalanine to see if this aromaticity increases its self-assembly property, increases its stability, and since we are increasing a new compound, they are also trying to see how the cytotoxicity has an effect. So, so the initial characterization, they have formed a gel and they have seen that they form the fibers. And so the storage modulus or the Ross modulus are all rheology properties when we see for the gel. So, what it actually says on a clear perspective is, it tells you how the storage of the gel is, whether it has viscosity property or a elastic nature. So, if the G prime is higher, it means the gel is more elastic in nature than that of a fluid nature. And these gels were very highly stable because of the indole moiety, and they have also done a cytotoxicity test in studying how the viability is different. So, only at very higher concentrations there is a reduction in the toxicity of the cells, saying that these can be used as a self-assembled system. And then this is a study where they have 5 different molecules. They have a peptide molecule, and in this peptide they wanted to have glucose into the structure, into the backbone. They wanted to see if adding a glucose has an effect in the thermal stability and biostability. Okay, so the major disadvantage of using all peptide systems are, these peptide systems are highly degradable. So, the amido bonds and all because of the enzymes that are present in our body, when you take a peptide self-assembled system inside our system, so what happens is, because of the environment they are highly degraded, but they are very highly compatible because they are taken as to be our own system. But so, to improve the thermal stability and the biostability, they have tried using glucose molecules, and glucose is known to have no cytotoxic effects. So, there are 5 different molecules that they have planned. So, you have the 1, 2, 3, 4, 5. First, napt is a naphthalene molecule towards your left, and then you have 2 phenylalanine rings that are conjugated, and then you have glucose, that is in the aspartate region they have modified to be glucose. So, 2 aspartate, 1 with each glucose. So, there are 5 different molecules each that have been changed to their glucose position. So, and the 5th one will have no glucose residue, only aspartate. So, that is to be taken as your control molecule. So, there is 4 molecules with glucose variations and the 5th one with no glucose variation. And so, they have summarized all the results in a table of how the thermal stability and the biostability of the system varies. So, the MGC here stands for the gelator weight percentage at the concentration at which the gel is being formed for all these peptide gels, and then the pH at which the gel is being formed, and then the width of the nanofibers in the fiber diameter. And the strain percent is the strain at which this gel becomes the solution. So, this also increases, or it depends based upon the addition or without the glucose. And then we have the biostability and the T gel. So, the biostability, when you see for the 5th one, is 0. So, here the biostability is after 24 hours of incubation in an enzyme fluid, they have checked what is the remaining amount of gel that is being there. So, in other cases you have a 48.58% or 29.48%, but without glucose there is 0% remaining, saying that the peptide hydrogel was completely degraded. So, glucose increases the biostability of this system. And then we also have a T gel, that is the gel temperature, the stability. So, the T gel of the hydrogels were studied where the glucose ones were higher than compared to that without the glucose one. So, this has a 53 degree at which it will degrade, whereas this has a very high thermal stability. And so, they have used 2 different gels, the first and the second gel, to study their cell culture activities. So, here they have done for NIH3T3 fibroblast cells and for the UX cells. So, the A and B, A is for the fibroblast cell line and B is for the endothelial cell line. Over a 5-day period, they have monitored how the cell proliferation works. So, the image that the green image that you see is when your cells are stained with fluorescein diacetate. It stains the cytoplasm, and that is the reason you have a green color image that is being seen. And then you have the C and D, where you have the actin staining that is happening. And then they have also calculated the cell density and the density percentage at which over a point of time to see how the proliferation has increased over a period of time. So, all this shows that these peptide gels can be used effectively for our tissue engineering applications for nice cell adhesion and then proliferation. So, this is another peptide self assembled molecule where they have a peptide amphiphile. 2 different peptide amphiphils have been taken, and then they have studied the initial characterizations for both the branched amphiphils, and then so they have tailored it with histidine, and these 2 different molecules have been studied separately and also in different ratios to see how whether mixing the peptide amphiphils has an effect in the tissue culture. So, here the figure that we see here is for 24, 72 and 96 hours in the fibroblast cell lines, where they have taken a specific ratio of both these polyamphiphils and then seen how the proliferation increases. So, this also shows that over a period of time they have a very good proliferations for these fibroblast cell lines. Here since we have seen different interactions, so that was how the hydrogen bonding interactions are. You also have ionic interactions based polymer gels for tissue engineering. So, here we have an ionic co-polyelectrolyte system where the polymers are modified to have a terminal ion. So, one is a polyanion and the other one is a polycation. So, when you mix these gel together, they form a cross-linked system, but these are not covalent cross-linked system, but they are intermolecular interactions. So, the purple color bubble that they say is where there is an ionic interactions between your 2 polymers. So, these are like the covalent bonds that are being formed holding the gels together. So, when then these put into the system, we see how they have an effect, whether the cross linking is effective or not. And then we have the host guest inclusion complexes. So, this is one of the most important molecule, the cyclodextrins. So, the cyclodextrins, as I have told, has a glycan nature. Here they have utilized 2 different ways of bonding them. One is they have modified the PEG terminal to be adenine and then the another PEG terminal to be thymine. As we have already discussed, adenine and thymine have a very nice complementary hydrogen bonding forming an AT pair. So, what happens is the PEG molecules when they bind together and form an hydrogen bonded AT pair. So, the PEG both are having an intermolecular interactions. So, these molecules then slide into the cyclodextrin core also to give a well known intermolecular interactions. So, this is how the gel is being formed, the image that shows. So, you have a solution and then mix them with cyclodextrin and then you see how the stability is better. And then this is a SEM image of how the gel looks, and here they have added doxorubicin, a cancer drug, and see how the release. So, but this can also, they have also done an in vivo study on rabbits to see how they can have an injectable effect of this gel on the rabbit. So, these are another small molecule gelators where we have alanine. Alanine is the core molecule. So, they have a small terminal modifications. Here the interesting part of the paper is that they have very small modifications to the terminal segments, showing that how small modifications or chemical modifications your system alter the self assembly pattern. So, you have the first molecule to be alanine hydrazide NH2. Whereas in the second one it is an alanine CAM, which is an C double bond O NH2, that is an amino bond. And then the other one is a protonated form of an hydrazide HYD plus. So, what happens when these are very small modifications when we do in a chemistry basis, but what happens when they move towards the self assembly process? So, the first one is the amino terminated one, the ALACAM molecule did not form a gel in any of the concentrations. So, that was ruled out. And then we have the alanine hydrazide and the protonated form of alanine hydrazide, which form gels at different weight percentage. But when they wanted to take them to be have an injectable system, these molecule gels are not that stable as a polymer gel, where they are very soft gels. So, they can be used as injectable gels. So, they have checked for the injectability of the gel and seen that alanine hydrazide alone has a very good injectable property in comparison that of the protonated hydrazide. So, only alanine hydrazide has been taken up for further applications of the drug loading and the drug release. So, this tells us that how small molecule differences have a greater impact when you go for the self assembly interactions. And here we have two different molecules coming into play. We have a peptide molecule and a polymer molecule. So, we have seen how a peptide assembly is, what are the advantages and what are the disadvantages of having a peptide assembly. So, the peptide amphiphils are most commonly used for the hydrogen bonding interactions, and they also have a anionic or a cationic interactions, and they have very tailorable bioactive properties. But polymers on the other hand are known for its stability, because they are high molecular weight compounds. They give the mechanical strength that is needed when you take them for a very nice tissue engineering scaffold that is to be formed. So, here what they are trying to say is there is a blend that is being formed between polymers and peptides. So, they can be both a covalent cross linking or a non-covalent interaction. Here we will be looking about only blending of these two giving a non-covalent interaction. So, when you see the peptides have a very nice hydrogen bonding giving an helical peptides or a beta sheets, whereas this will give you a high strength, and then that is how we combine to see how it has an effect. So, these are the different peptide molecules that have been synthesized. So, it is a long peptide amphiphil that they have synthesized with a long hydrophobic chain towards one end. And then they have an RGD peptide that is known to have a nice cell adhesion property that is being attached to that of your second peptide. And then the DGEA is also another integrin that can be used for cell adhesion properties. So, they wanted to compare what is the effect of having an RGD and what is the effect of having an DGEA. And then they are having this K3, that is a different functional group. So, the terminal functional group of OH and NH2 is varied between the E3 and the K3PA, and then we have all this is combined to that of your polymers. So, your polymers is a PEG DMA or a PEG dimethyl acrylate system. So, this we combine with that of the peptides and that of the polymers. So, when we have the SEM image coming into play, so you see the first column is the PEG molecule alone, which has only polymers that are being formed as gel. So, this does not give you a nice helical assembly or nanostructures that are being seen in your SEM. When we have the peptides having them in role, what happens? So, when you see the peptides having them in role, you see a nice helical or a nanofibrous structure in the gel form, because these small molecule assemblies, this is how they form. So, they form a bilayer, then go and then form an long nanofibrous gel, and this is how they form a big macromolecular gel kind of a thing. And to check for the stability, so the first advantage was to check whether the peptide has a role to play, and then the second one was to see if the addition of polymer has a role to play. So, the main aim here was to have an enhanced mechanical strength of adding a polymer. So, here we have PEGDMA at different percentages. When you see, you have it at 4%, 8% and at 12%, and when you see how the gels are. So, the slash lines give you only plain gels, and when you increase the G prime, you see that from the soft gel to the medium gel to a very stiff gel, how the G prime increases. So, the major soft, medium and stiffness is attributed because of the polymer component that we give. So, this is because of the 4%, 8 and the 12% addition, and the other, the G prime and G double prime, is because of the mixture of the gels that is being formed. And you also have the strain amplitude that they have studied, but the strain amplitude does not have a major effect when it comes to into play, because the most common feature that we are looking about is the stability or the tensile strength. That is what gives the G prime. And then we have the, they have done the cell culture studies for this. So, you have the PEG molecules and then without the PNFs. So, the PNFs are the peptide nanofibers. So, without and with the peptide nanofibers, how they act. So, here we have in the below image, C image, we have soft, medium and stiff. So, you have PEG molecules without the nanofibers and PEG molecules with the nanofibers at 3 different nanofibers. So, RGD and DGEA is known to have a cell adhesion properties. So, here we are trying to see which one has a better cell adhesion properties, whether the RGD peptide or the DGEA peptide, whereas your stability is given mostly because of your polymer, and then the peptide also acts as a self assembly property. Here when we look about the number of attached cells, So, the RGD molecule is very high when you compare with your soft gels and also in your medium and your stiff gels. But and then when we go for the spell spreading area, the RGD has a better role than in comparison to that of the DGA. So, when you see of all these integrin proteins which help in cell adhesion, when you compare them RGD and DGA, we say that RGD is much more effective than that of the DGA peptide. So, that is a common reason why people try conjugating RGD peptides to your polymers, to your any other gels materials, so that you have a very nice cell adhesion properties. And so, these are some of the examples that we have looked. So, this is how self assembly is and how what supramolecular interactions is. So, when you try understanding the basics of what a supramolecular interaction is, it will help you to design molecules for your applications. So, your large term application or your macromolecular aggregates are all depending upon your basic molecule, how your monomer assembles, what is the interaction the monomer has with its environment and with its own self. So, all this has a major role in designing molecules for your tissue engineering. Thank you.